I'm a grad student in Brad Peterson's lab, and today I have the true pleasure of introducing Steve Tettelbach, Dr. Steve Tettelbach. So Steve completed his undergrad at the University of Miami, then he went on to do a master's in fisheries ecology at the University of Washington, and he did his PhD at UConn on crustacean predation on Northern Bay Scallop. So he was a professor for 32 years. Up until recently, where he retired, and he's now a Shellfish Ecologist at Cornell Cooperative Extension. So throughout Steve's career, his research interests have been generally focused on understanding what drives populations to change, especially in a shifting climate. And he's applied his research findings to basically improve management of many different commercially important species, including bay scallops, whelk, and harpoons, among other things. So among Steve's wide array of research interests, for the past 40 years, he's worked on a lot of different questions surrounding bay scallops. And these have ranged from predator-prey interactions to all sorts of things influencing their survival, from habitat to water quality and that sort of thing. And he's really applied all of this knowledge to restoring bay scallop populations in the Connick Bay that were really on the brink of collapsing and disappearing. So this is one of the, arguably one of the most successful shellfish restoration projects ever, and I don't want to spoil too much. But he'll tell you about more about that in a moment. So I've known Steve for about a decade. I first met him when I was working out in Nantucket, and he was as well. And so I've had the opportunity to work with him on several projects since I've moved to Long Island, and also had the honor of having him as a professor on my committee. So Steve is truly an inspiring researcher, teacher, and an all-around great guy. So without anything else, take it away, Steve.
Well, thank you, Steve, for that wonderful introduction. I hope I can live up to some of that here. So, can everyone hear me okay? In the back? Yeah? Okay, good. So, just a spoiler alert, if you saw my talk in Southampton in February, it's not the same talk, but there's similarities. But what I want to do today is I'm going to talk about our bay scallop restoration work in the Peconics, but I want to focus more today on some of the ecological considerations. Some of the mechanics, some of the basic information that we really needed to be able to go at this restoration effort. Okay. So I'm going to focus more on these ecological questions in support of our bay scallop work. Oh yeah, that's much nicer. So here's our guy, or them actually. They're hermaphrodites, as you know. So bay scallops are they're a fascinating species. Obviously, I think so. I've worked on them for over 40 years now, but wonderful to eat, as many of you know. You may know them this way, or if you have the good fortune to go out scalloping, like some of us do every year, you may know them this way. Or if you're a shell collector, you may be astounded to recognize the incredible diversity of shell colors here. But this is an iconic species on Long Island, as it is in other parts of the Western Atlantic. And in the Peconics, it's really I have to use the word rabid. When the fishery comes along in November, everybody comes out in droves, and it's like this on Nantucket, of course. But so let's talk a little bit about the basic biology of scallops, because this obviously plays into the way that we went about doing restoration, and really the reason that we had to do restoration in the first place. So it's a very unusual life history. Usually live less than 2 years. They are only generally going to reproduce once in one season in their lifetime. So basically they reproduce generally at an age of about a year, and then they live for 7 months or more, and then they die before they actually spawn a second time. So it's a very strange life history. Friend on Nantucket, Steve Esterbrook, has actually looked at this and thinks it may be due to a single mutation with regard to the telomere length on the chromosome. So, but what it means in terms of the fishery is that these are going to die anyway, so they might as well get harvested. So people talk about over-harvesting scallops, but it really does not apply for the most cases. They're functional hermaphrodites. They're capable of self-fertilization, obviously external fertilization, and the larval cycle is generally regarded to last about 1 to 2 weeks. We usually use a figure of 2 weeks. So we go from the larvae in the upper right here through the post set. They will attach preferentially to eelgrass. This is their preferred habitat. Traditionally is viewed by some people as the only habitat where scallops are found. That is not true, and I'll talk more about that. But they like to get above the bottom, settle above the bottom, and then when they drop to the bottom, they'll attach to substrates like shell and so forth, and then the adult, as you see over here on the lower left. All right, so very rapid growth rate. In their first year, they grow at a rate of about 10 to 12 millimeters a month. So the area we're working in is the Peconics. The focus of our work is really in the bays that are highlighted here. So we've got Flanders Bay, Orient Harbor, Halleck Bay, and Northwest Harbor. So I'm going to be talking about those. And scallops have been harvested from Great South Bay, Moriches Bay, Shinnecock Bay over the years, but the bulk of the harvest historically has come from the Peconics. So the first part of our story is the good old days, what things were like up to 1985. And what you'll see here, these are the commercial landings for New York State, so this does include other areas besides the Peconics. So thousands of pounds of meats on the y-axis, and what you see is there's a lot of up and down. And again, remember that this is an animal that generally reproduces once. So if you get good recruitment one year, you get a good harvest, poor recruitment the next year. But tremendous variability, and that's normal for bay scallop stocks. The average harvest over this period was about 300,000 pounds of meats, and was believed to have employed about 400 to 600 baymen full-time during this period, and this represented probably about 2/3 of their annual income. Well, that all changed in 1985. The first of a series of brown tides appeared. This is one of the quintessential photos with the Atlantic Ocean to the south and I think Moriches Bay to the north here. So brown tide, first time it had ever been seen, 1985, and a lot of work, of course, has been done on that since that time, much of it by Chris Gobler and his group. But one of the first things that became apparent is that brown tide killed scallops, and it killed clams, and it killed oysters. But for scallops, it actually was a triple whammy. It led to starvation of adults and juveniles and presumably larvae, so populations declined directly. It interfered with recruitment. We did some work to show actually that scallops do spawn during the middle of a brown tide, but presumably the larvae don't survive because they're not able to feed effectively. But the third part of this trifecta was that the brown tide actually shaded out eelgrass, and eelgrass, again, being a preferred habitat. So 3 years in a row of brown tides, and if you have an animal that generally lives to only reproduce once, you end up with very low populations in very short order. So that's basically what happened, and within 3 years' time, bay scallops were essentially driven almost to extinction in the New York waters. So what I've done here is I've highlighted this period of the first 3 brown tides in the Peconics, 85, 86, 87, and you see what happened to the fishery. The harvest in 87 and 88 was 300 pounds, right? So 1/1000th of the average in the 30 years prior to that. We did have brown tides again in 91 and also 95. but what I want to do is talk about what happened right after the first brown tide. So because this fishery was so important, the baymen actually are the ones that started the restoration work. This is Pete Wenzel, is a very good friend of mine. I don't have a picture of Steve Watson, but these two guys were the ones that started scallop restoration in New York. And I actually came over to Long Island and started teaching in 1986, so right after the first brown tide. Some people would call that fortuitous, I guess, for my career, but it's just how it worked out. But so the baymen started this restoration work, and I got involved a couple years afterwards. So I just want to talk about a couple of theoretical considerations with regard to restoration before we get to our back to our story. So the basic idea behind restoration, of course, is to hopefully bring things back to better conditions, better population levels that existed previously, and ultimately the hope and the goal of most restoration efforts anyway is to attain some type of alternative stable state. So in this diagram, what you see is, this was pre-Agnes, Hurricane Agnes, Chesapeake Bay shellfish levels driven down to low levels, and then trying to get them back up. So restoration can be successful in some cases, but many cases it's not. But this is obviously what we're trying to do, is get it up to the next level. So population recovery can take place on its own, but in general it's believed that this has to occur from one or two conditions. Either the landscape has changed, for example, if brown tide disappears, or the population is altered directly. And of course, this is the way that most restoration efforts are conducted, is you try to increase the population directly. And ecological theory will say that once a population has reached an alternate stable state, it should remain there unless a larger perturbation comes along. So given that we are in the midst of doing restoration, I cannot comment whether or not we've actually reached an alternative stable state yet. We would have to stop that from happening, but we'll get to the results in a little bit. So what happened is that after the first three brown tides, restoration, as I said, started right away. And what you'll see here is that there was an uptick in landings. So there was some success here. A little brown tide in 91. For those of you who may remember, it lasted about a month, but we had a hurricane that year in October, and it knocked the brown tide out. And then all of a sudden in 1994, we had a harvest of 300,000 pounds, back to what the previous pre-brown tide levels were, and we're like, okay, we're done with restoration. We can go on with our lives, work on other things. And everybody was thrilled, of course. And then the next year, we had the worst brown tide of all during the entire time in the Peconics. And that reset the populations back to what they were. The harvest in 96 was 57 pounds for New York State. And I remember that distinctly because I was out scalloping on opening day, which I try to arrange my teaching schedule to be able to do every year. And I was out with Chris Smith, my good friend and colleague from Cornell Cooperative Extension, and we got like 2.5 pounds that year. And he turned to me and said, "We caught more scallops today than anybody in New York State." And I think we did. So the population got re-zeroed out. Okay. So now I'm going to focus on this period from 1995 up to 2007. Okay. Now, no brown tide. All right. We haven't had brown tide since 1995 in the Peconic Bays. Brown tides happen almost every year in the South Shore Bays, Great South Bay. All right, but no brown tide, and yet the landscape changed, but the populations did not recover. They stayed flatline. So the average annual harvest during this period, this 12-year period, was 3,000 pounds. So 1% of historical levels. So why didn't the populations recover? Well, we considered most of the suspects. We thought, okay, well maybe it's eelgrass's decline. A lot of people said, well, you can't be successful at restoration because there's there's very little eelgrass left. All right. We still know almost nothing about scallop diseases. I had done some work in 2000, 2001 in Great South Bay with Roger Newell, then at University of Maryland, and also worked with Chris Gobler to look at food quality and quantity might be a possible driver of this, and I'm going to talk a little bit more about that later. And then maybe some aspect of climate change. But what we came down to, our working hypothesis, was that the reason scallops did not recover on their own was that the densities and abundance of scallops had been reduced to such low levels that it affected the fertilization success, and therefore populations were recruitment limited. So when I taught at Southampton College of LIU, I would take my fisheries biology class out every fall, and we would go do quadrat counts in Northwest Harbor. And so these are data plotted from my class surveys. And what you see here is that we've got tremendously low population levels during this period. All right, so that backs up the hypothesis. So we were very fortunate to get 4 years of funding from Suffolk County, and a huge number of people have participated in this project. Some of you guys right here in the front row. And so what I'm going to talk about now is focus on the actual restoration program that we instituted. So these are co-authors on a number of papers, but there are many, many dozens and dozens of people that have been participating in this project. So the basic goal was to do large-scale plantings of hatchery-rich scallops at high densities, and I'm going to talk about the specifics as we go along here. Lots of monitoring, and the ultimate goal of this work has been to restore the fishery. Okay? Obviously, we need to restore population sizes, and to get to that, we had to boost larval recruitment. So that's the basic idea: boost larval recruitment, plant animals that are going to spawn and produce lots and lots of larvae, and then hopefully the population will rebuild. So, first thing we need to think about in bay scallop restoration: permits, right? So, Army Corps, Department of State, DEC, and so forth. But this process actually took a lot less time than I was expecting. It worked out pretty well. So we had to get animals, but the populations at the time we were doing this were so low that we really could not rely on transplant animals. So we had to rely on hatchery production, all right? So the initial hatchery production back in the 80s was actually done up in Maine, and then were brought down. So hopefully things were done in a kosher way with regard to the genetics, but obviously we tried to adhere to that in this 15 years of work here. So what we've done, there's two major approaches that we followed. One is to collect animals from natural populations, primarily through larval settlement, and then also raise animals in the hatchery. So we do this as part of our monitoring, is a collection of the scallop seeds. Again, this takes advantage of the scallop life history, that they will attach above bottom to basically anything. All right, so they love eelgrass, but they also will attach to macroalgae, and they will just as well attach to plastic. So that works out really well. So we put out these spat collectors, so mesh bags with another piece of mesh inside with floats on them to keep them upright, and we generally leave them out for about 6 weeks. But we've done this not only for monitoring, but also to collect animals to add to our hatchery production. And 1 year we did this in a serious way, and we caught about 250,000 scallops. Some other parts of the world, this is actually the way that they've gone about reestablishing and rebuilding populations. And the most iconic shellfish restoration program in the world in Japan with their scallops, this is the way that they did it. Hatchery production is done at Cornell Cooperative Extension's lab in Southold. And one of the things that we had to ask ourselves early on is, okay, so we're going to use hatchery animals. Are these basically equivalent to wild animals? And there's still, the jury is still out in a number of ways. But the first thing that we looked at was, okay, are these going to exert or invest as much of their energy in terms of reproduction as the wild scallops, and are they going to be spawning at the same time? And the answer to both questions was yes. So what we see here is hatchery animals are spawning. These were all transplanted into the same bay, Halleck Bay, spawning at the same time. The gonad index, which is the way that we look at reproduction, when you see a sharp drop in the gonad index, what that indicates is that the animals have spawned. So the mass of the gonad in relation to the rest of the tissues. So spawning occurred at the same time, gonad indexes are the same. So we felt good about this, that the animals are doing what we hope they're doing. Some recent work that one of my master's students at LIU posted looked at the shell strength of the scallops, and it turns out that the hatchery ones have much thinner shells, and so maybe more susceptible to predation, but that's a little bit of an aside. Okay, so we know where we're getting our animals from, so what size do we plant at? Okay, so obviously if you go the larval route, you can plant billions and billions of larvae for cheap. And that is one strategy that has been followed in some scallop restoration work, but it's had marginal success to date. But basically the overriding concern here is we have to minimize mortality. Right? So I had done my dissertation on predation on scallops by crabs in Connecticut. So when I got to New York, you know, it was all about crabs and basically getting them up to a size that we can minimize mortality. And of course, the primary cause of juvenile mortality for bivalves is in fact predation. So, and who eats scallops? Everything eats scallops. In the larval stage, at the adult stage, when I was working in Connecticut, you know, I got into the winter and I said, okay, the crabs have disappeared. I figured, you know, probably predation rate will drop down. Well, the seagulls showed up, and they watched me plant scallops, and bingo. So this is happening year round. So with regard to size of planting, this is from my dissertation, and this is a plot of predation intensity, a percent of planted scallops that were eaten versus scallop size here. All right, so very strong relationship here. Basically, when scallops get to a size of about 40 millimeters, they're fairly immune to most predators. All right. In Connecticut, the major predator that I found in my system, the Pequannock River in Groton, which is right next to the Avery Point campus, was the green crab, Personius manus. On Long Island, I still would say at the juvenile stage that crabs are the most important, but we do not see many green crabs out in most of the bays where we work anymore. Okay, so we know that we got to get these animals up to a large size, about 40 millimeters. Now this is way beyond the size that most hatcheries would produce scallops, but this is the approach that we follow both for our releases directly to the bottom, but also for ones that we plant in nets. So we tested after 1995. You know, we weren't sure if brown tide was going to come back. So the idea of getting more funding for restoration was kind of maybe not, but probably not. So we worked on a bunch of other things, and one of them was to look at, all right, what's the best way to overwinter scallops? So we looked at a whole bunch of different types of techniques. These are things that they've used in Florida successfully. These cages did not have much luck with that. So we settled on both these lantern nets here and then also free planting. I don't know if any of you have ever worked with pocket nets, but if you haven't, don't. They're a real pain. So these two methods, free planting and lantern nets, are what we focused on here. All right. Make sure I get this to see if this clicks. Okay, so this is a little video that Scott Hughes shot, Roy McNish from Cornell put together. So you'll notice that these are pretty small scallops, but we had a few 100,000 that we had to do something with, so you'll see. And I'm not shooting this, I'm on the boat. Scott's in. So one of the things that we've seen every time we planted scallops is corgis, scallop, come out of the woodwork. So this is one of the things that Steve Heck is working on now to see how important these are as predators of juvenile scallops. We think they're pretty darn important. So this is a fairly typical bottom that we like to plant on. Hard bottom, some macroalgae, but not too much macroalgae. Right? And you might be saying, well, why aren't we planting the eelgrass? Because there's no eelgrass within 15 miles of this spot. So high densities, all right? This is one of the things that we're going to focus on here as we go forward. Like I said, this is a much smaller size that we typically plant. We came back here about a month later, and there was very little evidence that any scallops had gotten planted on that site. All right? So we had to do something with it, and Steve could only use so many thousands of scallops. So that size does not really work for planting scallops. So this is some data from the early restoration efforts. Our initial plan at that point was to plant at about 10 per square meter. So I was still fixated on crab predation, attracting predators at high densities. There was a lot of work done with sea scallops up in Canada, and they showed that sea stars and crabs were attracted to planting. So we said, let's go at fairly low densities. And in some cases, we had pretty good overwintering survival. In some cases, the animals got hammered right away. Right? So what we ended up going to in this restoration effort, the last 15 years, is basically all modeled off what the Japanese did with their scallops there. So these are lantern nets. These are much larger than the ones we use. The scallops are much larger, but this is how they went about their incredibly successful scallop restoration work. This is just a quick shot of our long lines. You'll notice that these are in fact wonderful artificial reefs, and if you're interested in tunicates, come talk to me, because we have every tunicate in the Western Atlantic on our nets. So we plant these at about 35 scallops per tier. These are 5-tier lantern nets, and so these are obviously going to survive well, but they're also going to be at high density when it comes time to spawn. All right, so how many do we plant? Well, when we first wrote the proposal, I was like, yeah, let's try to plant 1 million, and Chris Smith said, no, let's plant 10 million. So we ended up somewhere in between. And if any of you know Chris, you'll know exactly how that is. But obviously, the idea is that the more you can plant, the better. And it really comes down to swamping the predators. All right? But when it comes to actually how many you're able to actually plant, it's, of course, going to come down to money and logistics. So we ended up stocking about 500,000 scallops into our lantern nets. All right? And part of that was the logistics. We wanted to plant more. The hatchery certainly produced more, but we just didn't have the space to do this. Right? So what kind of planting densities? Well, it's a trade-off between attracting predators and then also the idea of fertilization success. So after the early rounds of restoration work at 10 per square meter plantings, I started reading a lot of papers about recruitment limitation, fertilization success. And the literature is very rich in that regard. So we did a 180, and so we started going at planting at very high densities. In our lantern net system, we plant at about close to 200 per square meter, And again, large scallops, all right? We don't necessarily have to use as large scallops, but this is what the hatchery is geared up for. For our free planting directly to the bottom, we also plant at high densities. 75 to 100 is our target. And the whole idea is to get them, we want them close together so that when we spawn. So this was a paper we did based on direct field observations of scallop spawning in the wild. It was a quintessential nerd moment for me when I saw this, but it was really very cool. Had not been described before, but this is what we're after. And when we started, we did some preliminary work to this current project, and we planted at these high densities, and we did not see an influx of predators. Right? So we said, yeah, this is definitely the way we got to go. All right, so if you do the back of the envelope calculations here, stocking at 500,000 in the lantern nets at basically 100% fertilization success. That's what we've essentially used as our number from the models that are available, and the spawning of about 1 to 2 million eggs. So we're producing somewhere on the order of 250 to 500 billion larvae. Right? Got to swamp the predators. Right? Now going in, we didn't know how many we actually needed to be able to see a measurable effect. There have been other restoration efforts that had been successful on a short term. But you know, the literature will suggest, okay, do the calculations, what's the carrying capacity of the population, and we didn't do that. It's actually not an easy thing to do in a fluctuating environment like this. So we just went with what we could produce, and fortunately it worked out pretty well. So where do you plant, and how many sites do you go after? Well, we actually benefited from really excellent literature on the queen conch in the Caribbean by Alan Stoner and his group. And they actually saw that the most successful areas for planting were areas where the conch had historically been found. So we targeted areas that had been the best producers for scallops, Flanders Bay, Orient Harbor, and Northwest Harbor. So in the early days, we did plantings at tons of different sites, you know, a few thousand here, a few thousand there. And it comes down to the conservation biology question: single large or several small plantings. Right? So if you go large, then basically the idea is you've got more larvae in that larval cloud. And so this is basically the strategy that we adopted, is to go after this in a big way and try to basically put all our eggs in one basket and hope that the site we picked was a good one. Okay, so lots of things to think about with regard to where you do this. Free planting, we looked at all of these considerations here. We were most interested, of course, in predator abundance as well as the bottom characteristics. So we looked at these things in terms of the off-bottom structures. We had to obviously consider permitting issues, and then also navigation and so forth and so on. So what we ended up, well, a couple other things that were really important with regard to this. We were very fortunate to be able to use this larval dispersion model that was done right here at Stony Brook by Scott Siddell back in 1986 and his colleagues. And so they actually modeled where larvae would go if they were scallop larvae for the early restoration project, where they would go if they were released from a few points. So these are the 3 hotspot areas that we're targeting. And 2 weeks is the larval period that we focused on. So the red is as far as basically we're looking at larvae to go from these sites. Obviously Flanders Bay, they don't, what happens in Flanders Bay stays in Flanders Bay for the most part. But we were focusing initially on Orient Harbor. We figured that if brown tide had a chance to come back, that it was most likely going to happen in Flanders Bay. So we were thinking about Orient Harbor all along. So we wanted to see, all right, where are they going to go? Well, a lot of them stayed in the harbor. We actually did a larval drifter study with Joe Warren, and we're astounded to see that our citrus drifters went 3 miles outside of Orient Harbor on one outgoing tidal cycle, which kind of freaked us out. But Joe said, don't worry, they're going to slosh back in. And this has turned out to be a very good spot. But this was really crucial information to help us choose our sites. Eelgrass, as I said, is historically viewed as the most important habitat, but it's not all about eelgrass. Eelgrass has declined considerably over the years and continues to decline in the Peconic Bays. John Carroll, one of Brad Peterson's students and good friend and colleague of mine, and we have done a lot of work on codium, and codium actually serves a very important ecological function. So macroalgae seem to be covering for eelgrass, all right? And thinking about this, in talking to fishermen, Flanders Bay, which is one of the best producing areas for scallops, has not had eelgrass since maybe as far back as the 1970s, maybe even the 1950s, all right? And scallops have been there. So obviously there's a lot more going on here than just eelgrass. One of the other things we've been looking at is other habitats for scallops. So we've been looking a lot at Crepidula beds. There are scallops in each of these shots, some on tethers. We've been doing a lot of tethering work here. And this is proving to be a very important substrate for scallops to attach to. They actually will work themselves up inside the shells, get up in the cups here of these shells, and predation is definitely reduced. So this is a bridge between the off-bottom substrate and to a larger size where scallops can reach a refuge from predation in size. Another thing that we had to consider is this idea of winter burial. This is something I saw in Connecticut. I saw these scallops that were like this, and I'm like, what's going on? And I dug them up, and the tissues were, they were dead, but all the tissues were inside. So we did, this is my first grant in New York, was to look at this, and we found out that winter burial of scallops was very, very important. About 20% of scallops we planted got buried within about 10 days. So this is something we have to keep in mind with regard to habitat selection. And then we got to think about food. Okay, so yeah, maybe the reason scallops didn't recover was because there wasn't enough food or there's poor food quality. So a lot going on in this slide, but this is from the hard clam project that we did with Roger Newell. So what we've got here quickly is percent of chlorophyll, total chlorophyll broken down into size fractions. And we looked at 7 different sites for wild hard clams on Long Island and in Raritan Bay. And in some areas where there was a huge amount of chlorophyll, as in Great South Bay, it turns out that most of that food is not really suitable for hard clams. All right? So the red is not good clam food. All right? So we looked at that as part of our scallop work, but essentially did not see any evidence that either the quantity or quality of phytoplankton was limiting to scallops. So we ended up targeting Orient Harbor. This is the site of our primary spawner sanctuary. This is where we have our lantern nets. So if you drive to the Orient Point Ferry and you get past the causeway, look out and you'll see all the floats out there. That's us. And then we've also done free planting work in Halleck Bay and a few other places. Okay, so when do you plant? Well, most hatcheries spawn scallops in the spring. The seed don't reach the sizes that we want to plant until the fall, so we're constrained by that. One year we did a project where we were able to overwinter scallops with very, very high survival. This was done by the town of East Hampton Hatchery. And we did some spring plantings, and that actually worked out really well. So basically you can put them out a few weeks before they're due to spawn, and you pretty much have what you need to go with. But that's not something that generally can get done on a large scale. Okay, so let's get into the results of what we've done here. So tons of monitoring. We've done spat collectors as well as dive surveys throughout the Peconics here. 2 years of data before we actually started the restoration, and now we have 12 years afterwards. So spat collectors put out for 6 weeks. We cover the entire spawning period for scallops. This was the best day we had in 2007 early on. That year we caught about 1,000 spat for the entire year. And then by 2009 and 10, we were catching 100,000 scallop spat. All right, so this quickly escalated very quickly. And when we, this was our best day, about 35,000 scallops on one day. Okay, so clearly when you plot out the data, it proves that the populations were recruitment limited, which is exactly what we had hypothesized. Right? So we flooded the system with larvae. Okay, our dive surveys were to look at population densities, and at the same time we also do predator surveys. And this is actually where we've gotten a lot of really good data on the channel whelk fishery, which has been clobbered. So we're trying to work with Kim Account at DEC to try to get some regulations going because the populations have crashed. Okay, so this basically what you want to focus on is the number of circles and the size of the circles here. These are changes in mean density over an 8-year period. Obviously, we have more data from this, but this is what we published in the 2015 paper. So when we started out, you know, we would do our transect surveys, and if we saw one scallop in a 50-meter line, you know, we were excited. And then we got to a point where one dive, it took me 2.5 hours to do one transect, 50-meter transect. There were so many scallops. So populations increased, obviously, in numbers, but also in the areas where they're found. I talked to Ken Holman, the owner of Ron's Seafood in Cutchogue, a couple years ago, and he said, yeah, they're in all the old places, and they are. So now the populations they're not just based on what we're doing, but the natural populations have recovered to such a point where they're really sustaining themselves on their own, okay? For the most part, although we believe that our continued involvement is an important buffer. Okay, so the bottom line for us was what's going on with the fishery. So these are DEC data, and what we have plotted here is the 12 years leading up to our restoration work, so averaging about 3,000 pounds, and then things took off. So we thought that maybe we reached an alternate stable state at about 40,000 pounds afterwards, but we were delighted to see that it kept going. So the last 2 years have been the highest scallop landings in the Peconics since 1994, 2 years in a row. So we're up over 100,000 pounds. Now again, historically the average was 300,000 pounds. So do we have more work to do? Yes. Can things get better? Yes. But things have come a long, long way. Economically, this has been a real boon to the baymen, which I'll talk about in a second. So we looked at, okay, what are the possible drivers of this? So we looked at before and after with regard to all the usual suspects here: temperature, chlorophyll, nitrogen, rainfall, salinity. Not even close to any significant difference between the before and after. We looked at wind patterns. We looked at really everything we could think of. All right. So we kind of put together the slide as possible reasons for why scallop populations increased, and we looked at all of these different factors. So weak arguments are in red, orange are a little fuzzier, and then really the only strong argument that comes out of this is that this increase in population size and fisheries is driven by our restoration work. And the thing that really clinched it for us was a model that we put together. This was based on a study that was done by Lundquist and Botsford. And what we did for this was we plotted out on the x-axis the index of fertilized egg production. So what this takes into account is scallop densities that we have obtained in the field directly, and we plug those densities into the model, Lundquist and Botsford model, to calculate percent fertilization. So we're able to come up with numbers of fertilized eggs for each of the systems that we looked at, and then we plotted this against our actual larval recruitment that we saw in our spat bags, and very, very strong relationship here. So at least for the initial stages of restoration, there's a very strong correlation between stock recruitment relationship. Okay, historically in fisheries this is not the case for most situations, but we saw this very, very strong signal. So very important economic benefits to local baymen. There are more baymen harvesting now. Jobs and economic benefits trickle down from this as well. And then if you look at how much money that we've actually gotten for this project, which is a little bit above $4 million now, but the increase in landings above the baseline in the 12 years before we started the restoration is almost double that now, directly to baymen. And then when you add in the economic multipliers, it goes way beyond that. So, take-home messages. We were fortunate enough to get funding for 4 years initially, and then we've been very fortunate to have gotten renewed every year since then, right? So other restoration efforts that, did this for a year or two, they saw some benefit, but then it kind of just went away and dissolved into the background. So initially we didn't see much of a change, but then things really popped after a few years. So basically building up that spawning biomass is what was the key to this. We went all in on our one site. We wanted to get as many larvae in that larval cloud. We didn't do the tracking of larval clouds, but this is actually what they did in Japan as part of their efforts. And you see this giant cloud of larvae drifting around, and then when it's time to set, boom. And so we went all in on this, and fortunately we picked a good spot in Marine Harbor. Really extensive monitoring is essential, and so many restoration programs have not been able to do this, largely because of funding, but this has been a huge component of our work, and it's been very important. The best habitats for planting, and again, with almost no eelgrass, we're looking at macroalgae, but not too much macroalgae. So we think there's issues with DO levels getting too low in the summer. So we're going after moderate densities, greater than 2 meters, less chance of burial, and fairly hard bottoms. And Crepidula is a really good substrate for scallops. So we are actually targeting those areas now. All right, so here's our Where's Waldo shot, if you want to try to count these up again. But the point I want to make here is that the recovery to this point of the scallop populations has occurred with almost no eelgrass. Okay? And so macroalgae, incredibly important, as our work has been showing. And we didn't know that this was going to be possible, but it's happening without eelgrass. And there's even the heretical idea that eelgrass may be a larval sink. Now, the reason I say this is that we have 24 sites that we regularly monitor. Okay? Yeah, right. What? We have 24 sites that we regularly monitor, and one of them has eelgrass. All right, and it's off the north side of Shelter Island, Hay Beach area of Orion Harbor, and this is an area where we've seen tremendous sets of scallops over the year. But when you go back in the spring, they're all gone. Okay, and there's a lot of burial that happens here. So right now, eelgrass beds on Long Island are primarily in far eastern reaches. They are in areas of fairly high current, and the scallops, when they set there, actually do not survive very well for the most part. Now, Caitlin may argue with me, but that's what we've been seeing so far. So it's not anything that we expected at this point, but so that was definitely a surprise. So sustained restoration is likely necessary to be able to rebuild these stocks, and right now we think that we are able to help buffer those big fluctuations in recruitment that happen naturally in scallop populations. But right now it's the natural populations that are driving the show. And again, we don't know if we've reached a stable state because we're still doing this, but we're going to keep doing this as long as we can. And tons of people to thank. Huge effort over the years by a ton of organizations, many people from Stony Brook as well. So thank you to all. And that's my story. So. Oh. Nope, not that one. Okay. Any questions? Yes, Glenn.
Thanks very much for your talk. Some encouraging news.
Thank you.
I was very interested in this, the effect, this failure of reproduction in low density.
Yep.
In hermaphrodite really says that self-fertilization is not very important. Are there conditions where it is?
Yeah, we actually do think that self-fertilization may be important at extremely low densities. There was some work done a few years ago where they looked at doing self-fertilization crosses, and they found that in the F1 generation there was really no deleterious effect, but in the F2 there was. So it may be a way for a bottleneck to get through that period. One of my students right now, Marissa Velasquez, is looking at whether scallops aggregate in preparation for spawning. We think they do. We've seen it over the years, even at really low densities. Obviously, they can swim, but and it does look like they do in preparation for spawning. So that's an important thing that you have to add into the modeling of this. But yeah, so self-fertilization may be important at very low densities, and that may be what got the populations through those really lean years. Initially. Yeah. Any other questions? Niels?
So 1994, it seems there was a huge recruitment, even though you started off with a really low standings.
Yep.
Would that not suggest that in principle the species could be sustained itself now, because now it's even much higher? What was going on in 94?
Yeah, that's a very good question. There was some genetic work done on the early restoration efforts. Maureen Krause, who was then at Southampton, did some genetic work to show that the restoration was contributing, certainly at least in part, to that increase in population size. But 94, I won't even begin to suggest that what restoration we were doing is what caused that. That just came out of the blue. So that was a big question. Why did that happen, and why didn't it happen after in the 12 years? So we were wondering, are there some other fundamental ecological conditions that have changed in the bays afterwards? There were other communities of invertebrates. I remember Raoul Welker at Southampton saying, yeah, all the sponges, that he would see in the normal places, they weren't there. So there may have been some other ecological conditions, and we really can't begin to suggest what those might have been. So yeah, that 94 is a big question mark. In the literature, of course, that there are other examples where populations will go from very low to all the way up here. But what I would say in response to that is that, well, if you have a much larger brood stock available, then that certainly can drive and more consistently be able to contribute to the larval production. So yeah, I can't answer exactly what happened in 94. Yes.
So one thing I want to point out is that relationship with brown tide. It seems that part of the story here is this continued success because of lack of brown tide that's happening within the system.
Oh, sure, yeah.
And what would you attribute to the lack of that brown tide?
Well, I remember talking to Chris Goldberg about this, and maybe I shouldn't say this, but Chris assured me that brown tide wasn't going to come back after 95, so he's been right so far. But one of the things that, and I was just talking to Boston and Manuel about this, is so Crepidula fornica is very abundant in the conics. In the lab, it's been shown to not only eat brown tide, but actually to grow on brown tide. And in Great South Bay, there is virtually no Crepidula left. And I talked to old-time bayman, Marty Burns, the town of Islip, and there used to be tons of Crepidula back in the 70s, and now there is virtually none. We're doing a project now. Brad Peterson, John Carroll, and I got funded to do a project there, and there's no Crepidula crepidula there. So that there's no substrate in most places that we've looked. And so that might be part of the reason why, and obviously, Bob's work, the shellfish aren't there. So there's nothing really to control these HABs in Great South Bay. But Connex, crepidula's there, and then now scallops have increased in numbers, and so they're able to help control the brown tide more certainly than they did. So, but again, just to reiterate, there was that 12-year period with no brown tide where the scallops did not recover. So it's taken off since then, and no brown tide as well. But didn't happen on its own.
And one other comment, I'm kind of disheartened to hear about the relationship with eelgrass site. One of the things,
Oh, I know. I get a few people up in arms when I say this.
Well, one of the sort of drivers in terms of public relationship. I mean, people love scallops, and so you try to get them behind eelgrass restoration or at least efforts for conservation, saying, oh, that was a part of the history
Sure.
of this habitat. So.
Well, what I will say is we've actually done a lot of work looking at eelgrass as the settlement substrate versus macroalgae. And based on our work, I would definitely say that eelgrass is the most important habitat. Okay, and the reason I say that is because scallops are able to attach and stay attached to eelgrass above the bottom for much longer than on macroalgal species. Right. So the longer the scallops can stay above the bottom, away from many of their predators, not all, the better. So eelgrass is able to accomplish that. All right. But so I'm not saying that eelgrass is not good. Don't read that at all. It's the best substrate. But right now, where it's located, it's not really the place that scallops are going to survive the winter.
Yeah, most of that's really open water sites.
Yeah.
Probably not.
Yeah.
The
Yeah.
other conditions.
I mean, if eelgrass were in the inner bays, of course we'd be all over that. But it's not. It's not.
As an
Yeah.
evolutionary strategy, it would seem to me that being able to reproduce once in your lifetime is a pretty risky endeavor.
Yeah, I mean.
Has anybody thought of maybe some genetic modification that might induce them to spawn ice?
Well, very good question. We were actually just talking about that this morning. So the answer is yes. Steve Estabrooks on Nantucket actually did some work, and I don't know if he's published it yet, but he was looking at species Argopecten purpuratus from Chile, which is very closely related, but it lives to 10 years old. So he actually was going to do crosses between these, not to suggest that would be a viable thing to do. But why do they not die right after spawning? Okay, I mean that's a very common strategy amongst insects and many other types of animals. But there is a component of the population where there are some animals that do live and spawn at age 2, and these are ones that actually are a fall spawn scallop initially, and they set late. They're very small going into the winter, and the work that had been done up on Nantucket, Valhall, has shown that about half of those scallops spawn in their first summer, but the other half don't spawn until they reach year 2. So that turns out to be a really important buffer to the population, because now instead of just a single year class, you've got basically, more than one, not 2 full year classes spawning. And what we've seen is that those actually really are very important to scallop populations here, and work done in North Carolina has also shown the same thing down there. So yes, that would be wonderful for scallop populations to have that additional buffer of another year class. Yeah. So, but in terms of genetically doing that, I suppose it's possible, but other than crossing with Chilean scallops, no one's really looked at that yet. Any other questions? Yes, Bob.
Maybe along the same line of genetics, I'm doing the parallel with infectious diseases. After a severe mortality, usually survivors are better fit to survive future events. I understand that the restoration was initiated with Maine scallops, but in future years, and I mean the following years, also the main source was continuously used, or you started?
No, that was actually in the 80s. The main scallops were used in the 80s because there weren't any hatcheries on Long Island that were actually growing scallops at the time. But no, for these 14 years, we've been using scallops from Long Island, and yeah, wild caught, natural populations, and trying to go along with all the good genetic guidelines. That have been all. Yeah. See.
Were the scallops at the main hatchery? Were those scallops that were brought up there from Long Island?
No, I think they came from Cape Cod, actually. I mean, it's like we were talking about moving shellfish around. Obviously, it's been a lot with scallops. Over the years, there were actually introductions of scallops. I think in the 70s from Massachusetts into Great South Bay. Actually, not huge numbers, but some. So it's a little muddy, but the ones in Maine, I think, I mean, they're not native to Maine naturally, so they were brought up there from Massachusetts. I think. So, but that was really the only place we could get them.
Any more questions? All right, so
Okay, thank you very much.
again.
Thank you.
And we'll have lunch in the exhibit room.